In this video, we will discuss about LDL metabolism. Lipoprotein LDL is formed from VLDL, very low density lipoprotein. Normally, VLDL is synthesized from liver by packing with all the endogenously synthesized lipids. That means both triglycerides and also cholesterol. Initially, the triglyceride and cholesterol ratio within the VLDL will be 5:1, 5 parts of triglycerides and 1 part of cholesterol. So when this VLDL is metabolized by the peripheral tissues, they will be taking up the triglycerides. Okay, the triglycerides will be lost and ApoC will be lost and they will be converted to IDL, where the ratio between triglyceride and cholesterol reduces to 1:1. Okay, so VLDL, so during its catabolism, it will lose its triglycerides to the peripheral tissues, ApoC back to the HDL, and they will be forming intermediate density lipoprotein. Now from this intermediate density lipoprotein, if you remove further triglyceride molecules, okay, and ApoE protein, it will be converted to cholesterol rich ApoB100 containing LDL molecule. This is how LDL is synthesized from VLDL. The peripheral tissues which converts VLDL to IDL, it uses an enzyme called as lipoprotein lipase, which will be present on the surface, capillaries present on the surface of all these peripheral tissues. So they will hydrolyze the triglycerides in the VLDL and will be taking up all their contents. Next, from IDL, it will be converted to LDL by an enzyme called as hepatic lipase, mainly the hepatic lipase enzyme. They will bind the LDL, they will take the triglycerides and convert it into cholesterol rich LDL molecule. Okay, so this pathway in which one lipoprotein is converted to other lipoprotein is called as lipoprotein cascade pathway. This is through this pathway only LDL molecule is synthesized. Now this synthesized LDL, which is rich in cholesterol, will be delivering its content to the peripheral tissues. How it delivers, we will see in detail now. So all these peripheral tissues, most of the peripheral tissues, they will be having a specific receptor to take this LDL molecule, which is called as LDL receptors. These LDL receptors are always clustered in a specific region on the membrane called as clathrin coated pits. Okay, so these are the clathrin coated pits where the LDL receptors will be clustered. So when the LDL molecule comes in contact with these receptors, the ApoB100 of LDL molecule, they will bind with these receptors and results in endocytosis. That means the receptor along with the entire LDL molecule will be taken up by the endocytic vesicles. Now this vesicle, which is carrying the LDL and LDL receptor, will fuse with the lysosomes, and within the lysosomes they will be catabolized and the cholesterol will be released into the tissues. After taking up the cholesterol, the LDL receptors will be recycled back and it will be sent to the membrane to catch further LDL molecules. This is how cholesterol from LDL will be taken up by the peripheral tissues. Okay, so LDL molecule as a whole, first it binds to the LDL receptors with the help of ApoB100. After binding the receptors along with the LDL molecule as a whole, it will be engulfed. Okay, by endocytosis, it will be taken in. They will fuse with the lysosomes. The lysosomal enzymes, they will be destroying, they will be catabolizing this thing. They will release the receptors back to the tissues and free cholesterol will be taken up by the tissues. Okay, so now this free cholesterol has two different fates within the cell. One is it can be used for any biological functions. Cholesterol has lot of functions. Okay, so it is needed for cell membrane synthesis. It is needed for steroid hormone synthesis. It is needed for vitamin D synthesis. It is needed for bile salts, bile acids of formation. All these functions are there. Okay, so depending upon the cell, it will be used for that particular function. After serving all these functions, if there is excess cholesterol, it will be stored within the tissues as cholesterol esters with the help of an enzyme called as acyl cholesterol acyl transferase. That means the enzyme will be transferring the fatty acid to the cholesterol. That is why it is called as acyl cholesterol acyl transferase. So from a fatty acid group will be transferred to cholesterol to form cholesterol esters. So when this cholesterol esters starts accumulating within the tissue have a plan, or they will have a process to inhibit the further intake of cholesterol into the tissues. So how the further entry of cholesterol into the tissues inhibited means by three ways. The first one is the production of LDL receptors will be reduced. Okay, so if LDL receptors are not produced, further LDL molecule cannot be taken up. Second, it produces a protein called as PCSK9 protein. Okay, what is the function of this PCSK9 means? During LDL metabolism, we saw that after within the lysosomes, after releasing the cholesterol, the LDL receptors will be recycled back to the membrane for further uptake. Okay, so this recycling of LDL receptors back to the membrane will be inhibited by this PCSK9 protein. Okay, so they will be inhibiting it. So further uptake of cholesterol will be reduced. On third way is it decreases the HMG-CoA reductase enzyme so that the endogenous synthesis of cholesterol will be stopped. So by all these mechanisms, the cells will prevent further accumulation of cholesterol within the tissues. Okay, so if it is not accumulating, okay, suppose if it is not taking up the LDL, what will happen to that LDL molecule? They will be accumulating in the blood. So this excess LDL accumulating in the blood will be oxidized by the free radicals and it will be converted to oxidized LDL or some other modified LDLs. So these modified LDLs, they will be taken up by macrophages through the scavenger receptor A. This is an alternate way of LDL catabolism. Okay, normally it will be taken up by the peripheral tissues using LDL receptors, but when there is enough amount of cholesterol within the cell, the peripheral cells will not take the LDL. So in such conditions, the LDL will be taken up by the macrophages using scavenger receptor A. Okay, so when this LDL enters into the macrophages gets transformed into foam cells, and that foam cells in turn leads to an most deleterious process called as atherosclerosis. Okay, and this in turn can complicate as myocardial infarction and also stroke. That is the reason we call this LDL cholesterol as a bad cholesterol. Understood? So cholesterol, if LDL, if it is in excess, it can lead to atherosclerosis when it is taken up by the macrophages. To prevent this and to reduce the LDL cholesterol, we have various therapeutic options. The first and most important among them is to reduce the cholesterol intake. Okay, so cholesterol intake has to be reduced. It is not only cholesterol, even if you take excess fatty acids or excess glucose in your diet, that can also be, the acetyl-CoA formed from it can also be utilized for cholesterol synthesis. So it is better to reduce the calorie intake along with the cholesterol intake. Apart from reducing the exogenous source, you should also inhibit the endogenous synthesis. How it can be done means by using HMG-CoA reductase inhibitors like statins. There are various statins available which by competitive inhibition can inhibit the HMG-CoA reductase enzyme and they can decrease the endogenous cholesterol synthesis. Apart from that, we have also a few newer drugs which can decrease the LDL production and it can prevent the risk of atherosclerosis. They include evolocumab. It is actually a monoclonal antibody which is produced against PCSK9 protein. Okay, so under metabolism, we discussed that this PCSK9 will help in preventing the recycling of receptors back to the cell membrane. Okay, so when that is inhibited, what will happen? Receptors can be recycled back. So if receptors are recycled back, it can be taken up by the peripheral tissues. So what will happen? LDL will not accumulate in the blood, that cannot be oxidized and that cannot enter into macrophages and it cannot lead to atherosclerosis. Okay, so all these things can be prevented by using this PCSK9 inhibitor, which is a monoclonal antibody, evolocumab. Okay, so apart from that, we also have a drug called as lomitabide, which can inhibit the protein microsomal triglyceride transfer protein. So this microsomal triglyceride transfer protein is the one which is very much needed for VLDL synthesis. So when VLDL is not produced, which is actually the precursor of LDL, even the LDL production will be affected and LDL production will be decreased, and thus it can help in decreasing the risk of atherosclerosis. And finally, the last drug is mipomycin, which is actually inhibits the ApoB synthesis. ApoB is the one LDL. It has only one apoprotein, which is nothing but ApoB100. Okay, so if the synthesis of that is inhibited, parallelly, automatically lipoprotein LDL production also will be inhibited, and the risk of atherosclerosis will be decreased. Okay, so that's it about LDL metabolism. To sum up, LDL is produced mainly from VLDL. Okay, so VLDL, after losing its triglycerides and the other lipoproteins except ApoB, they will be converted into LDL molecule, which will be having cholesterol and ApoB100 alone. Okay, so now this LDL will be donating its cholesterol to the peripheral tissues through LDL receptor mediated endocytosis process. Okay, that's it. Thank you.